What's up, nerdlings? I'm back to present to you gene regulation in eukaryotes. So eukaryotes have a wide variety of ways that they can actually regulate their gene expression. So it's because they actually have that temporal separation between transcription and translation. They also have this thing called a nucleus that prokaryotic cells do not. There are 5, count them, 1, 2, 3, 4, and 5 main gene regulation methods. Those include DNA access control, or epigenetics. I have another lecture over that coming out shortly. I love the field of epigenetics, so that will be exciting. Pre-transcriptional control, post-transcriptional control, pre-translational control, and post-translational control. We'll see if I get tongue-tied during this lecture. Chances are yes. So DNA access control. All cells have the exact same DNA content, but not all cells need to express every single gene. So again, for example, heart muscle cells shouldn't be able to produce adrenaline. So different types of cells will permanently or temporarily be inactivated or downregulated / turned off to control that gene expression. And they do this by manipulating how DNA is actually stored in the cell. So they do this in 2 methods. We have euchromatin and heterochromatin. Euchromatin is whenever we have the unwinding of DNA, which does allow for transcription to occur. Heterochromatin is when DNA is super tightly wound around histone proteins, and we call that methylation. And this is whenever those transcription factors can't get on to where the DNA needs to be transcribed and actually transcribe it into a messenger RNA strand. So, in eukaryotes, DNA is stored by being wrapped around histone proteins. Now, depending on how loose or tight that is going to regulate gene transcription. So, these proteins can either be moved closer together or further apart to control access to the genes that are wrapped around them, stored in DNA. So heterochromatin is whenever DNA is tightly wound around histone proteins, and those are packed very tightly together. Genes are unavailable for transcription, so they're super tightly packed together. Now in euchromatin, this is when DNA is loosely wound around histone proteins. There's a lot of space in between, and this does allow for transcription to occur. So switching between euchromatin and heterochromatin. This allows cells to control access to genes by changing which regions the DNA are arranged as either euchromatin, meaning they can be transcribed, or heterochromatin, meaning they're tightly coiled around those histone proteins and cannot be transcribed or upregulated. So this is done by adding small chemical groups to the histone proteins. So we have acetyl groups, and those spread the histone groups out. This can cause the formation of euchromatin, and this makes genes available for transcription to occur. We can also methylate groups, and this causes histone proteins to pack tightly together, and it causes the formation of what we call heterochromatin. This can silence genes because genes are unavailable for transcription to occur. So pre-transcriptional control. So eukaryotes require transcription factors to actually bind to the promoter region before RNA polymerase can bind and begin transcription. So controlling the expression of those transcription factors leads to gene regulation itself. So if the transcription factors aren't made, the RNA polymerase can't drive the gene. So pre-transcriptional control allows for the differential gene expression in cells. So certain cell types will permanently downregulate or silence genes for certain transcription factors. Using that process I just talked about, methylation, by methylating it, it remains in that heterochromatin state where the DNA is tightly wound around those histone proteins. These transcription factors will be permanently unavailable, so RNA polymerase will never be able to bind to the promoters of those genes. In a post, or meaning after transcription has occurred control, messenger RNA requires post-transcriptional modifications to leave the nucleus for translation. So we have to remove the introns and this happens at the spliceosome. So remember, introns remain inside the cell. Exons, the spliceosome splices the introns and exons together. The exons get to exit the nucleus and actually be upregulated when they get to that ribosome. So the removal of introns by the spliceosome, as well as the addition of the 5' cap by enzymes and addition of the poly A tail. Those are basically two little keys that messenger RNA has to use in order to exit the nucleus so they don't get degraded by enzymes. So messenger RNA can't leave without those modifications. And eukaryotes can control access to the spliceosome and enzymes that lead to those modifications, which allows for gene regulation. In pre-translational control, the messenger RNA has now left the nucleus. It relies on non-coding RNA called microRNA. MicroRNA sequences are transcribed and then they're chopped into smaller fragments by what we call a dicer protein. These microRNA fragments then bind to proteins. If the microRNA protein complex encounters a messenger RNA with a complementary sequence to the miRNA, it will base pair with the messenger RNA, and this can target the messenger RNA for degradation, meaning it recognized that there was something wrong with it, so it's going to break it apart. This also prevents ribosomes from translating that messenger RNA strand into a fully functional protein. So either way, translation does not occur, so again, that gene is not expressed. In post-translational control, we have destruction of unnecessary proteins. These proteins will be tagged with a molecule called ubiquitin. It's added to certain amino acids in the protein sequence, and this tags them for degradation, basically saying, whoa, 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 there's something wrong with that guy over there. We don't need him going out into the cell and being upregulated and wreaking havoc. So they're then transported to a proteasome, which breaks the protein into fragments, and amino acids are then recycled and reused. Very responsible. So I hope that was helpful for you, learning about eukaryotic gene regulation. Stay tuned for my film on epigenetics, and I hope to see you all soon. Stay nerdy till next time.